Imagine your immune system synthesizing literal, actual household bleach just to dissolve a bacterial invader.
Yeah, or, your liver acting as this ruthless, highly guarded customs agent, just intercepting and dismantling foreign chemicals before they can ever even reach your brain.
Right, because we tend to think of human health in these macro terms, like a fever, a cough, a broken bone. But underneath all of that is a microscopic, just fiercely bustling chemical metropolis.
It is. It's a metropolis where every single reaction is highly regulated, entirely interdependent, and operating at lightning speed. So when a patient sits in front of you with a complex set of
Because
symptoms,
you're not just looking at the symptom.
exactly, what you're really looking at is a metabolic traffic jam.
And that is exactly what we are exploring today on this deep dive. There is this simple pencil sketch tucked into the opening pages of our source material. It's a drawing of a medical resident.
They have a stethoscope draped around their neck, a pen in their pocket, and a look of just deep, exhausted focus.
We've all been there. And our mission today is to map out the entire blueprint of medical biochemistry, not as like a list of pathways to memorize, but as the ultimate tool for that resident. Because every biochemical pathway you understand directly translates into a better clinical decision for the patient.
It's foundational. You can't fix the engine without knowing how it runs.
Okay, let's unpack this, because before you can figure out how that patient gets sick, you have to understand how their body is built right down to the molecular studs and drywall.
Yeah, you have to start with the physical components. We're talking about amino acids linking together through peptide bonds to fold into functional proteins.
The building blocks.
Exactly. We have carbohydrates snapping together via glycosidic bonds, which are essentially dehydration reactions, right, that lock sugars into energy-storing chains. And then you have lipids.
Which are so cool because of how they behave in water.
Right. Because lipids have hydrophobic tails, they naturally self-assemble into bilayers in water, physically creating the boundary of every single cell in your body.
So these are the bricks?
These are the bricks. But the architect dictating how those bricks are laid is the molecular basis of inheritance. So DNA, RNA, and protein synthesis.
But the sheer scale of this architectural blueprint is completely counterintuitive to me. The human body has about 20,500 genes. When I first read that, I had to stop and push back, like, we only have 20,500 genes.
It sounds low, doesn't it?
It's wild. That is roughly twice as many as a tiny fruit fly, Drosophila melanogaster. How do you go from an insect buzzing around a rotting banana to a human being with a cerebral cortex capable of doing calculus with just double the genetic code?
What's fascinating here is that the complexity of an organism does not come from the sheer volume of genes. It comes from the highly intricate ways those genes interact, are spliced, and are regulated. It's not just a static parts list.
It's what you do with the parts.
All right. We have moved entirely into the systemic omics approach. Genomics is your list of 20,500 parts, but then you have transcriptomics, which is how that list is read and edited.
And then proteomics, right?
Yes, proteomics, which translates to roughly 30,000 distinct proteins. Because one single gene can be spliced to create multiple different protein variants.
And then it just completely explodes into metabolomics.
Exactly. Those 30,000 proteins interact to create a staggering 52,000 metabolites circulating in your body right now.
52,000. That is a massive jump.
It is. And managing all this chaos are discoveries like microRNAs. There are over 1,800 of these short, non-coding RNA strands.
And they don't even build proteins, right?
No, they don't build proteins at all. Instead, they act as microscopic silencers. They physically bind to messenger RNA and block it from being translated, effectively turning genes off at highly specific moments.
It is a brilliant system. But I have to admit, when you look at a chart of human metabolism for the first time, it looks exactly like the London tube map. I mean, it is this chaotic web of lines, arrows, and intersecting stations that just seems impossible to navigate.
It really looks like spaghetti on a page initially. But just like the tube map, it is highly navigable once you know the major transfer stations.
Right.
Understanding this map is the very foundation of evidence-based medicine. When you know the routes, you can identify clinical biomarkers. If a specific metabolite is piling up in the blood, you know exactly which enzymatic train station, so to speak, has been shut down.
And then you can pinpoint the disease.
Precisely.
Which brings us to the engine itself. We have the map, we have the molecular bricks, but static molecules do nothing without power. The continuous provision of energy metabolism is what keeps the system alive.
And the core of this is oxidative metabolism,
Hmm.
which is essentially a tightly controlled low-temperature combustion reaction inside our cells.
A slow burn.
Yeah, and that combustion happens inside the mitochondria, primarily through the tricarboxylic acid cycle, the TCA cycle. It is basically a microscopic turbine.
Taking the fuel from our food.
Right. It takes the metabolic fuels derived from your gastrointestinal tract, the glucose from carbohydrates, the fatty acids from lipids, and strips electrons away from their carbon backbones.
And those electrons are the key.
They are. Those electrons are then passed down the electron transport chain, pumping protons across a membrane to create a battery-like gradient. When those protons flow back, they physically turn an enzyme that synthesizes ATP, the universal energy currency of your cells.
And oxygen is the final acceptor for those electrons, which is why we breathe.
Exactly.
But this engine requires a flawless integration of nutrition and metabolism. When that fuel supply gets disrupted or, chronically overwhelmed by our modern diets, we see the biggest global health crises of our time: obesity, atherosclerotic cardiovascular disease, and diabetes mellitus.
Diabetes is the textbook example of a metabolic derailment. It really highlights the profound difference between the fasting state and the postprandial or fed state.
Because the body handles those so differently.
Right. In a healthy fed state, your pancreas releases insulin. Insulin binds to a receptor on your cells, triggering a cascade that pushes glucose transporters to the cell surface, pulling sugar out of the blood. But in type 2 diabetes, chronic oversupply of fuel and inflammation lead to insulin resistance.
But mechanistically, what actually is insulin resistance? Because we always hear the cells stop responding, but what is physically happening?
Well, at a biochemical level, the insulin receptor becomes desensitized. Due to chronic high levels of insulin, the cell actually internalizes its own receptors.
It hides them.
Yeah, it pulls the biochemical mailbox off the front porch, essentially. The insulin is knocking, but there is no receptor there to receive the signal.
Wow.
So the glucose stays trapped in the bloodstream, forcing the entire metabolic map to reroute. The liver starts inappropriately making new glucose, thinking the cells are starving.
Because the signal isn't getting through.
Exactly. And the chronic high sugar damages blood vessels, leading to the long-term vascular complications of diabetes.
I was reading about oxygen transport in the context of these metabolic pathways, and I came across something that just blew my mind regarding sickle cell disease. It turns out that some adults with sickle cell are treated by stimulating the production of fetal hemoglobin like baby hemoglobin. How does a fetal protein fix an adult's red blood cells?
Oh, it comes down to structural biochemistry, which is so elegant. Adult hemoglobin is made of 2 alpha and 2 beta protein chains. In sickle cell disease, a single amino acid mutation in the beta chain substitutes a water-loving amino acid for a hydrophobic, water-fearing one.
A tiny typo in the DNA.
Just one typo. And when that adult hemoglobin drops off its oxygen, that hydrophobic patch is exposed, and it desperately tries to bind to other hydrophobic patches to hide from the water.
So they clump together.
Right. The hemoglobin molecules stick together, polymerizing into long, rigid fibers that physically warp the red blood cell into that sickle shape.
And those sickled cells jam up the capillaries, causing agonizing pain and tissue death.
Precisely. But fetal hemoglobin, or HbS, doesn't use beta chains at all. It uses gamma chains. And gamma chains do not have that hydrophobic mutation. If you can use a drug to turn the fetal hemoglobin gene back on in an adult so that their blood contains about 20% HbF, it completely disrupts the polymerization process.
So it breaks up the clumps.
Yeah, the HbF molecules act as blockades, preventing the adult hemoglobin from forming those rigid fibers. Furthermore, fetal hemoglobin binds oxygen more tightly because it has a reduced Bohr effect.
The Bohr effect, that is the mechanism where hemoglobin releases oxygen in the tissues, right?
Correct. As your tissues metabolize, they produce carbon dioxide and protons, which lowers the local pH. That acidic environment physically changes the shape of the adult hemoglobin molecule, forcing it to drop its oxygen exactly where it is needed most.
Like dropping off a package when it reads the right zip code.
Great analogy. Fetal hemoglobin is just less sensitive to this. So, by understanding this exact molecular shape change, a resident can understand why a specific pharmacology improves a patient's breathing and saves their tissues from infarction.
It is brilliant. So we have the engine running, we have oxygen flowing, but an engine cannot run unsupervised. With all this energy coursing through 52,000 metabolites, the body's tissues have to communicate to maintain homeostasis. We have to look at cellular signaling.
The environment outside a cell is constantly changing, and the cell must adapt. It does this via signal transduction, You have cell surface receptors, the most famous being G-protein-coupled receptors, or GPCRs.
Those are the ones that weave through the membrane, right?
Yes, these are proteins that weave back and forth across the cell membrane 7 times. When a hormone, like adrenaline, binds to the outside of the receptor, the entire protein twists and changes its physical conformation.
And that twist on the outside wakes up the G protein sitting on the inside of the cell membrane.
Yes. The G protein swaps out a dormant molecule for an active one, breaks apart, and slides across the inside of the membrane to activate an effector enzyme.
It's like a molecular relay race.
It really is. That enzyme then pumps out thousands of second messengers, like cyclic AMP or calcium ions. This is how you amplify a signal. One single hormone molecule on the outside results in tens of thousands of second messengers flooding the inside of the cell.
Turning on enzymes, changing gene expression, or sparking an action potential.
Exactly.
Which brings up a critical communication pathway we haven't touched on yet, neurochemistry. The brain is the ultimate command center, but it operates in a highly exclusive environment due to the blood-brain barrier.
The blood-brain barrier is a fascinating anatomical and biochemical defense. The endothelial cells lining the capillaries in your brain are fused together with extremely tight junctions, and they are surrounded by the fatty feet of astrocytes.
So it's not like the rest of the body.
Not at all. Unlike in your arm, where nutrients can easily slip between capillary cells, in the brain, molecules are forced to go through the cell membranes.
Meaning if a molecule isn't highly lipid-soluble or if it doesn't have a specific active transporter door built for it, it is just not getting into the brain.
Exactly. This protects the brain's delicate neurotransmitter balance. Neurotransmitters are synthesized right there in the neurons. When a signal arrives, they are dumped into the synaptic cleft, the tiny gap between neurons, where they bind to receptors.
Triggering that second messenger cascade we just talked about.
Right, and then they are rapidly cleared away or dismantled by enzymes so the signal doesn't endlessly loop.
Here's where it gets really interesting to me, looking at how tissues specialize. We have the brain locked away behind its barrier. We have the lungs and kidneys constantly managing our acid-base balance using the bicarbonate buffer system. But then you have the liver. The anatomical placement of the liver is so central.
It gets everything first.
Right. It receives venous blood directly from the intestines via the portal vein. I like to think of the liver as the body's strict, heavily armed customs border.
It is the perfect analogy. Everything you absorb through your gastrointestinal tract, every carbohydrate, every lipid, every nutrient, and every single foreign substance, including therapeutic drugs, must pass through the liver's customs checkpoints before it is allowed to enter the systemic circulation.
The liver processes the raw materials. It orchestrates the acute phase reaction during inflammation by churning out defense proteins, and most importantly, it detoxifies.
If we connect this to the bigger picture, this customs border dictates the entirety of clinical pharmacology. Because the liver intercepts everything, it attempts to metabolize and break down a large portion of a drug before it ever reaches your brain or your heart.
Which is first-pass metabolism.
Yes. The liver uses an arsenal of enzymes, notably the cytochrome P450 system, to physically stick hydroxyl groups onto toxic, fat-soluble drugs, forcing them to become water-soluble so your kidneys can pee them out.
And this is why drug dosages matter so much. If you take an oral medication, the doctor knows the liver is going to destroy, say, 40% of it immediately, so they have to give you a high enough dose that the remaining 60% actually does the job.
Precisely. And if those detoxification pathways are overwhelmed, disaster strikes. Take a common acetaminophen overdose. Normally, the liver safely neutralizes the drug.
In an overdose.
In an overdose, the primary pathways are saturated. The liver is forced to use an alternative pathway that generates a highly toxic intermediate. Unless the body has enough of an antioxidant called glutathione to neutralize it, that toxic metabolite violently binds to liver proteins,
causing massive, fatal, drug-induced liver injury.
Yes.
It is an incredible burden for one organ to carry. But the liver doesn't work in isolation. Let's trace a causal pathway from the liver to another highly specialized tissue bone. The liver starts the process of activating vitamin D, right?
It does. The liver takes raw vitamin D and adds a hydroxyl group to it. But it is still inactive. The liver ships it through the blood to the kidneys, which add one more hydroxyl group, creating the fully active hormone, 1,25-dihydroxyvitamin D.
And that hormone travels to your intestines to maximize calcium absorption, which then directly impacts your bones.
Because bone isn't just structural scaffolding. It is a highly active metabolic bank salt for calcium. The skeleton contains 99% of your body's calcium in the form of hydroxyapatite.
And hydroxyapatite isn't dead stone, right?
No, it is a living, crystallizing mineral lattice. It's constantly being dissolved and rebuilt. You have osteoclasts, which secrete acid to melt the crystal and release calcium into the blood when levels are low.
And the builders.
Osteoblasts, which lay down new protein matrix to rebuild the crystal. This dynamic equilibrium is exquisitely controlled by vitamin D and parathyroid hormone.
And supporting that skeletal frame is skeletal muscle. The mechanism of muscle contraction is just mesmerizing, the sliding filament model.
It's pure biomechanics.
You have these thick myosin protein filaments that physically grab onto thin actin filaments, burn an ATP molecule for energy, and ratchet themselves forward, sliding past each other to shorten the muscle. But the clinical takeaway here is the biochemical reality that exercise is medicine.
It is arguably the most potent medicine we have. As we age, or in states of chronic disease, we lose muscle mass, which is a condition called sarcopenia. Because muscle is the primary consumer of glucose, losing muscle means losing your metabolic sink, paving the way for insulin resistance.
But when a muscle contracts, it does something incredible. Muscle contraction actually forces those GLUT4 glucose transporters to the cell surface, independent of insulin.
Right, you don't even need insulin.
The mere mechanical act of exercising pulls sugar out of the blood. It is a direct biochemical intervention that treats diabetes and maintains metabolic function during aging.
Which naturally brings us to how this entire system defends itself and how it ages. We have built the map, powered the engine, and specialized the tissues. Let's look at the blood, which is our diagnostic window into this metropolis. When a resident draws blood, they have to know the difference between plasma and serum.
Let's break that down mechanistically. You draw blood into a tube, spin it in a centrifuge to push the heavy cells to the bottom. The yellowish liquid on top is what we're talking about.
Yes. If you draw that blood into a tube containing an anticoagulant, the liquid on top is plasma. It contains water, electrolytes, and all the circulating proteins, including a massive soluble protein called fibrinogen.
But if there's no anticoagulant?
If you draw the blood into a tube with no anticoagulant and let it sit, the coagulation cascade activates. Enzymes cleave that soluble fibrinogen, turning it into an insoluble sticky web of fibrin that traps cells and forms a solid clot.
So the clear liquid left over.
The clear liquid that remains after the clot forms is serum. So serum is just plasma that has been stripped of its clotting factors.
And circulating in that fluid is our immune defense immunoglobulins hunting for antigens. But sometimes the threat isn't an external bacteria, it's an internal rebellion. Let's talk about the cell cycle and cancer.
Cells are not supposed to divide endlessly. The cell cycle has rigorous biochemical checkpoints, governed by proteins like p53 and the retinoblastoma protein.
The inspectors.
The inspectors, exactly. Before a cell copies its DNA, these proteins inspect the system. If the DNA is damaged, p53 halts the cycle. If it can't be fixed, p53 triggers apoptosis.
Which isn't just cell death, right?
No, it is a highly choreographed, programmed dismantling of the cell so it doesn't spill its toxic contents and cause massive inflammation.
But if a mutation knocks out p53, that checkpoint fails. The cell ignores the stop signals, ignores the apoptosis command, and divides uncontrollably. That subversion of physiologic growth is what creates a tumor. Cancer is fundamentally a failure of metabolic regulation.
And alongside the risk of cellular mutation comes the broader process of aging. If you look at a Gompertz plot, which graphs the mortality rates of a species over time, you see that the probability of death increases exponentially as we age.
But the why is fiercely debated. Is it biological, meaning our DNA has a genetically programmed obsolescence, like a biological clock running out? Or is it chemical?
Well, the most prominent chemical theory is the free radical theory of aging, which ties into the mitochondrial theory. The idea is that as those microscopic mitochondrial turbines in our cells burn oxygen to make ATP, they occasionally drop an electron.
Just a little leak in the system.
Right? And that loose electron latches onto oxygen, creating a reactive oxygen species, or ROS.
And a free radical is incredibly dangerous because it has an unpaired electron. It is wildly unstable. It will violently tear an electron away from the nearest lipid membrane, protein, or strand of DNA to stabilize itself,
causing a chain reaction of oxidative damage. The theory suggests that over decades, this cumulative oxidative stress damages our mitochondria so badly that energy production plummets, creating a vicious cycle of decline.
But wait, I have to ask, are these reactive oxygen species always the biochemical villains causing aging and disease, or does the body actually use them?
That is a brilliant transition, because while ROS damage our tissues over time, They're not entirely evil. In fact, our immune system deliberately weaponizes them. When a macrophage, a type of white blood cell, engulfs an invading bacteria, it traps it in a cellular bubble. Then it initiates a sequence called the respiratory burst.
Weaponized oxygen.
Literally.
Yeah.
The macrophage consumes a massive spike of oxygen. It uses an enzyme called NADPH oxidase to steal an electron and force it onto the oxygen, deliberately creating that dangerous superoxide radical.
Oh, wait.
Another enzyme quickly converts that superoxide into hydrogen peroxide.
Okay, hydrogen peroxide is toxic enough.
But it goes one step further. A third enzyme, myeloperoxidase, takes that hydrogen peroxide and uses it to oxidize chloride ions, creating hypochlorous acid.
Hypochlorous acid? Wait, isn't that the exact chemical formula for household bleach?
Yes. Your immune cells are generating localized microscopic bursts of dilute bleach to dissolve the cell walls of invading bacteria.
That is unreal.
This raises an important question for modern medicine. How do we balance giving patients antioxidants to soak up free radicals and prevent aging without accidentally disarming the oxidative weapons our immune system desperately needs to fight off infections? It is a delicate biochemical tightrope.
Wow. Okay, we have covered an immense amount of territory today. From 52,000 metabolites mapping out our molecular architecture, to the liver acting as a heavily guarded customs checkpoint, down to macrophages synthesizing literal bleach to protect us. Biochemistry isn't just a list of enzymes you memorize for a board exam. It is a highly integrated causal system.
It connects everything.
So, what does this all mean? It means that understanding this blueprint, knowing how and why these reactions happen, is the absolute foundation of clinical reasoning.
Exactly. When you understand the mechanism, you don't have to guess. To reinforce this for the students and young professionals listening, I want to leave you with a specific active learning exercise based on our deep dive.
Let's hear it.
Imagine you are reviewing a patient's chart. They suffered severe lower limb injuries in a traffic accident. Now, 5 days later, their blood panels show that their C-reactive protein, or CRP levels, are massively elevated, while their serum albumin, normally the most abundant protein in the blood, is significantly decreased.
CRP is soaring, albumin is plummeting.
Based on what we discussed today, take a moment to deduce what this specific pattern of plasma proteins tells you about the patient's current metabolic state. Jot down the biochemical mechanisms that explain these laboratory results. Think about our liver customs border. How and why does the liver reprioritize its protein synthesis factories during an acute phase response to systemic inflammation?
That is a phenomenal prompt to test your mechanistic thinking. I highly encourage you to map out that causal chain. Thank you for joining us on this deep dive today. The next time you are on the ward or even just looking at that sketch of the exhausted resident, remember, you have the intricate map of the human engine in your head. Use it to find the source of the traffic jam.